Now let us talk about C4 cycle. What appears to be comparatively difficult is much easier, because over here what you have to deal with is only the C4 acid that would be formed. Rest everything is to be done by C3 cycle only. First thing that you have to keep in mind that the ultimate carbon fixation would be done by Kelvin cycle only, whether it is C3 cycle or it is C4 cycle. The only difference that lies is that the first formed chemical compound by fixation of carbon dioxide in this particular cycle is a C4 acid, that is oxaloacetic acid. Please remember it, that the C4 acid that is being formed is oxaloacetate. Now what happens over here is, as I told you in the beginning when we talked about the Kranz anatomy in the previous lesson, we had discussed that there are two types of cells, mesophyll cell and bundle sheath cell. Inside the bundle sheath cell, the Kelvin cycle is taking place because over here O2 is absent. Rubisco is present, O2 is absent, so there is no chance of photorespiration taking place. Now, in order to make that sure that over here only carbon dioxide comes instead of oxygen, because it avoids the entry of air totally, that is the reason why it is in the form of a wreath, so that there is no air that comes inside. What you see over here is these are two different cells. This is the mesophyll cell, this is the bundle sheath cell. This represents the outer cell wall. These are the plasmodesmatal connections which are present. This is plasmodesmata, the passage that has been represented over here. You know that plasmodesmata is present in the plant cells as the intercellular connections that is present. So we have plasmodesmata over here. We have the plasmodesmatal connections through which the transport would take place. So if we have to represent C4 cycle, we have the process of carboxylation, then we have the process of transport. So this first step is the process of carboxylation. Then we have the second stage, that is of the transport. The third stage is of the decarboxylation, and the fourth one is regeneration. Over here again we have transport of the C3 acid. This won't be regeneration. We have the fourth stage, that is the regeneration. So this is all simply the main cycle which is responsible for fixation of carbon dioxide happens to be Kelvin cycle only. The difference lies what is taking place in the mesophyll cells. As I told you, the mesophyll cells over here lack rubisco. Instead they have phosphoenol pyruvate. In place of RUBP, it would be phosphoenol pyruvate, which would combine with the carbon dioxide. The atmospheric carbon dioxide comes into the mesophyll cells. It combines with the water. It forms this HCO3. This HCO3, it combines with phosphoenol pyruvate, which is again a 3-carbon compound. On addition of this HCO3 to phosphoenol pyruvate in the presence of enzyme peptase. Okay, or we can say phosphoenol pyruvate carboxylase that comes into the scenario, it leads to the formation of a C4 acid. Okay, now this C4 acid is a result of carboxylation process that is taking place inside the mesophyll cell. Once this C4 acid, which fixes the atmospheric carbon dioxide, is being formed, it would be transported to the bundle sheath cells. Once it enters the bundle sheath cells, it is going to get rid of its carbon dioxide that it had brought from the atmosphere as a result of the first reaction that we discussed. It is going to get rid of the carbon dioxide and become C3 acid again. The C3 acid would be transported back for regeneration in the mesophyll cell, and the carbon dioxide that is released, that is again in the gaseous form, would enter the Kelvin cycle where we have rubisco and RUBP which would come into the scenario. So this is all about the C4 cycle. It is quite simple. You can keep in mind that this part, the bundle sheath cells are performing the C3 cycle only, but the mesophyll cells have to make sure that they provide the carbon dioxide, because no atmospheric exchange can take place inside the bundle sheath cells. The carbon dioxide would be provided by some acid that would be formed in the mesophyll cell. So the acid that would be formed, that would be oxaloacetate, which takes up carbon dioxide in the presence of phosphoenol pyruvate carboxylase enzyme when it combines with a 3-carbon compound called phosphoenol pyruvate. Now this is what basically C4 cycle is. Now what we have to keep in our mind, which we have to discuss in detail, little bit of it, that how this C4 cycle takes place, because we are primarily dealing with this part. This was different from the other part. Rest was simple, that a carbon dioxide is being released, Kelvin cycle is taking place. We have discussed about Kelvin cycle. What we have to discuss is that what is the difference, what are the stages of difference, and if there is any difference between C3 and C4. As I told you, because of Cran's anatomy, because of getting rid of the wasteful process of photorespiration, of course this C4 cycle is going to be far more productive in terms of photosynthetic efficiency. But we have to see how many ATPs are being used, because in terms of ATP, we understand what is the input of the particular cycle. Now, what you see is that we have phosphoenol pyruvate. That was the beginning. This phosphoenol pyruvate, on addition of carbon dioxide, I am not representing it as CO3, simply on addition of CO2, it becomes oxaloacetate or oxaloacetic acid, which can change to aspartate as well. We don't get into details of that. Simply this oxaloacetate, in the presence of malate dehydrogenase, that is the enzyme, it gets converted to malate. Now this malate enters the Kelvin cycle. Okay, this malate would go, it would get rid of its CO2 and enter the Kelvin cycle. For the regeneration of phosphoenol pyruvate, here we are going to have this malate, because it gets rid of the CO2 it had, it would become phosphoenol pyruvate. In this process, what we are going to have is that an ATP would be utilized to form an AMP and give rise to 2 inorganic phosphates. Alright, so we have for one carbon dioxide molecule, we have over here the fixation that is taking place. You know that 3 ATPs would be used in this Kelvin cycle, and there are 2 more ATPs. Okay, this ATP is forming AMP, so in a way we are having 2 ATPs being used. So we have an input of 5 ATPs per carbon dioxide molecule that is being fixed. Okay, now this 5 ATP would amount to totally, for a glucose molecule, because there are 6 carbon dioxides which are fixed, we have 30 ATPs which are being used. Please remember this. This you have to remember. In this process, what happens is an NADPH plus H positive gets converted to NADP positive, but whatever is being utilized over here, that would be regenerated in this process. So we have NADP positive that gets reduced, and we have NADP plus H positive. So we don't calculate NADPHs because they are getting regenerated. What is being utilized over here, that is getting regenerated over here. But what is to be noticed, that the 2 ATPs would be used additionally to the 3 ATPs which were utilized in the Kelvin cycle. So in total, for one carbon dioxide molecule, we have 5 ATP molecules, which amounts to a total of 30 ATP molecules which are utilized in a C4 cycle for the formation of one glucose molecule. So you have to keep this thing in mind. Now what comes to mind is that it is using so much of ATPs, so how it is a useful process? See, mostly these C4 plants, they are found in tropical regions, and in tropical regions energy is not at all limiting. Okay, you have so much of sunlight that is coming. Generation of ATPs is going to be abundant. So utilizing more ATPs and not wasting the ATPs is more important, because not wasting the carbon dioxide molecules, because carbon dioxide is always going to be a limiting factor for photosynthesis. You know the atmospheric content of the carbon dioxide, how much less it is in comparison to oxygen. So it doesn't matter if 30 ATPs are being used. So there are certain students who have a doubt that it is utilizing 30 ATP molecules, how can it be better than the C3? Because we are ultimately concerned about the productivity in terms of glucose. We have C4 plants mostly growing in tropics, and in tropics energy is never a restricting element. What is restricting everywhere is the carbon dioxide availability. So this particular C4 cycle, which takes a different passage, and by having a 4 carbon compound formed in the beginning and a Kranz anatomy coming in hand for help, has an upper hand over C3 cycle in terms of productivity. So that is why we say that C4 plants are always more productive as compared to the C3 plants. Now I hope by the end of this lesson you're quite aware what C4 cycle is all about. The primary cycle for the fixation of carbon dioxide here also stays C3 only. The only difference is that the C3 arrangement is made in such a way that no oxygen comes, and for that the plant has enabled the carbon dioxide fixation into another compound that makes sure that oxygen does not come due to the anatomy which is present in the C4 plants. And for that anatomy we have talked about in the previous lesson. So this is all about C4 cycle.